So basically, you guys have an exam in 1 week. So for the internet students, on your Blackboard account, you will now see, not the face-to-face students, just the internet students, you will see a message was activated today, because it's a week before the exam, that says you need to click on this to register for whether you're going to take the exam with the face-to-face students, because then I know how many paper copies to bring, and or you will have, you're denoting that you're going to take it at one of the testing centers. You have to, face-to-face students cannot take it at the testing center, only the internet students. Internet students, you have to make your own appointment. There is a link on that message that comes up to take you to the testing center website so you can make your appointment to take your exam there. For internet students, the exam opens Wednesday 8 a.m. until Thursday 1 p.m. That way, in case there's an emergency, you could run into my classroom and take it with 1 o'clock, room 106, Meyer Health Science, 1 to 2:15. If you get done early, you're done. You don't have to stay till 2:15. It's designed to be a 50-minute exam, 50. You guys actually have 75 minutes to take the exam. Make sure that if you're coming, you have your student ID, because I provide the Scantron for you. You just need your student ID and a pencil. You need to put your student ID on the Scantron, and if you don't, it delays your Scantron from getting graded. And or if you put the wrong number on it, or you don't bubble it in properly, et cetera, it delays your exam from getting graded, and you might end up waiting upwards of a week to find out what your test score was. So make sure you bring both of those next Thursday. Hopefully today we will finish Exam 1 material. I will put a line across the notes so you'll know exactly where Exam 1 material ends. And then Tuesday, when we come back to class, we should be starting on Exam 2 material. I understand if it might be a little light, because people don't always want to move on to new material when they're trying to just focus on what's going to be on the exam. But just make sure that if you do something like that, that you still go back and listen to the lecture video. Okay, so I just kind of rewound a little bit back to when we'll review real quick on enzymes. So enzyme function, we said that they're used to build or break things down. It's very specific, right? One enzyme, which is a protein molecule, one protein enzyme only works, has the proper shape for one reaction, nothing else, right? And I don't know if I mentioned it, I did say that it speeds up chemical reactions, but it speeds them up to 1 billion times faster. That's where I gave the example of lifting the pencil really quick and it takes me 1 second to do it, versus taking me 12 days to slowly raise my hand up. So it speeds it up that fast, 1 billion times faster. And I'm not making, it's not like a billion kajillion, like billion is the real number, not like my 7-year-old, like, Mom, it's a gazillion faster, okay? So, and I mentioned things like enzymes that we know end in ase, like protease, lipase, carbohydrates. I verbally said that to you guys last time. I didn't handwrite it out. So I just wanted to add that in real quick. And let's see, what else do I want to say? We wrote out the names. You guys know that it's covalently bound backbone on the DNA and RNA molecule of the sugar ribose or deoxyribose and a phosphate group. Those are the backbone of your DNA or RNA molecule, and the nitrogenous base, adenine, guanine, cytosine, thymine, or uracil. Thymine's for DNA, uracil's for RNA. Those nitrogenous bases, hydrogen bond across would be like the ladder rungs, right? RNA is single-stranded, DNA is double-stranded. But RNA, for a short time, will hydrogen bond to the DNA molecule that it is looking at as its template. So you have complementary base pairing. You always know who can pair up with who. And RNA does for just a short time, where we unzip the DNA molecule, RNA gets made, RNA comes off, DNA can zip back up when you don't need to read off your little message anymore, right? So just a quick review on that. We said that DNA is the instructions for everything that you could possibly need to make for you. So I call it like the Joy of Cooking cookbook, every single recipe possible. But we don't need every single recipe every single time. We just want to take off the one little message, the messenger RNA, the one little recipe card. We're going to print that off, and that will, so DNA is found in the nucleus, RNA is made in the nucleus, but then it goes where? goes out to the cytoplasm to find a ribosome. We're going to talk about ribosomal subunits in just a moment. And you're going to see that we mentioned some of these things now, and then we build on it and add to it and keep giving you more information. So I hope that some of me repeating some of the stuff is not redundant for you, but that it helps go, oh yeah, I remember, and now I'm adding to my story, and now I realize why she said that 5 million times. So that was, last thing we talked about was ATP, which we're going to mention again in a moment, adenosine triphosphate, 3 phosphates attached to, oh look, there's a ribose sugar, and oh look, there's adenine, one of the nitrogenous bases that we also use for DNA and RNA. So now we're jumping to chapter 4, biology of the cell. I talked briefly about the cartoon picture of a cell, and again, this is just a representation of the cell. Not all cells look like this, but most cells are going to have the nucleus usually kind of centrally located. They're going to have rough endoplasmic reticulum and smooth endoplasmic reticulum and so on and so forth. They're going to have mitochondria in it. They're going to have a Golgi apparatus. They may have microvilli. They may not. They may have cilia on it. They may not. Not too many of our cells have a flagella on it, but we know one in particular that does, a sperm cell, right? And then vesicles are like little lipid membrane bound, because everything's like a phospholipid bilayer, right? So lipid bound, and sometimes these vesicles will carry digestive enzymes in it, and we'll give it a new name like lysosome. And then sometimes they are packaged things that they can then transport in or out of the cell. So this is where I talked about exocytosis and endocytosis, right? We're going to talk about that again towards the end of today's lecture. So then we said the cell was the basic structural and functional unit of life, right? And then our cells will come together to form tissues. Tissues will come together to form organs. Organs will come and maybe have organ systems. And this is more 228, but I'm like peppering it in there now. So it'll be like, oh, okay. So trillions of cells in over 250 basic types. So we actually have about 75 trillion cells. Again, not a made up trillion. It's a real number, right? 75 trillion cells in your body. It just kind of boggles your mind when you think of it that way. We said that they can have varying shapes and sizes. So for example, a muscle cell, which was the first one here, I said is multinucleated. So it doesn't just have one nucleus. It has multiple nucleus in a skeletal muscle. And skeletal muscle will look different than smooth muscle, which looks different than cardiac muscle. And you guys are going to look at that if you haven't already. You're definitely going to look at that this week as you guys are doing histology in lab, okay? And the length, for example, of one muscle cell, so if I'm talking about like a bicep muscle, one muscle cell will be the length of your whole bicep muscle. Okay, so that's a fairly long cell. But then we also talked about a nerve cell that could be up to 3 feet long, like your sciatic nerve, and go all the way down to your big toe. And depending on how tall you are, I'm kind of short, so mine's not as long as yours, maybe. And then red blood cells don't have a nucleus. So they actually start off with a nucleus, but then it gets ejected. They don't have a nucleus. So it's one of the few cells, like the only cell that we really talk about, that has no nucleus in it. So that's kind of different. But what does that tell you about that cell if it doesn't have a nucleus? It won't have DNA in it. If it doesn't have the DNA, it doesn't have blueprints to make another one of itself. So you're going to have to make red blood cells from somewhere else. It can't replicate itself. Versus like your skin cells have a nucleus and they can replicate themselves. So if you bump up against something, get a scratch, have a surgical procedure where something's shaved off, you can make another cell, an identical cell. Not quite like identical twins, but essentially an identical cell. It's going to know how to be a skin cell, okay, an epithelial cell. And if we couldn't replicate those, you'd start to look like Swiss cheese, especially as a little kid because of all the bumps and bruises and bangs that you end up doing and scratches and stuff. You'd look like Swiss cheese. You wouldn't be able to fix the holes you've scraped out of yourself, right? So we have to be able to do that. DNA is going to allow us to do that. Nucleus contains the DNA. So we'll talk about that some more. So general structure, we said the nucleus is the control center of the cell. It's large because, again, you guys could see this under the microscope, right? You may not be able to see, probably you shouldn't be able to see. You can't see the rough ER or the smooth ER. You can't see the Golgi apparatus. You can't see the mitochondria. Okay, but you can see a nucleus, and then you can tell that there is cytoplasm between the nucleus and the cell membrane. And that's about all you can see with the regular microscope. Okay, you'd have to use something I said like an electron microscope. We'll see a picture in a minute of what a grainy electron microscope looks like. And essentially, they just had to take electrons and bombard the cell structure with it so that you could then see what it would look like. So it's centrally located usually, but not always, but typically, and it contains genetic material such as DNA, right? We also have a plasma membrane, which is the outer limiting boundary of the cell. And then we said we also have cytoplasm. Okay, let's skip ahead of my notes. So a cytoplasm, that's a general term for everything in between the plasma membrane and the nucleus. So just a generic term, right? Now, in cytoplasm, we have a combination of cytosol, organelles, and something called inclusion. So I just ended on the last lecture with this information. So cytosol is the intracellular fluid. Intra meaning within versus the term extracellular means fluid that's outside of a cell. Because remember I told you guys we have water outside the cell, water inside the cell, and we have that phospholipid bilayer that's preventing too much of that water from getting in or out of the cell, or things that should be inside a cell from getting out of the cell, or things outside of the cell from getting in. That's the job of the plasma membrane. So cytosol is like all the stuff, the liquidy portion, the intracellular fluid that makes up the foundation of cytoplasm. The majority of cytosol is made out of water, so that's the majority component of cytosol. But it also has ions in it. Those are charged particles, right? We talked about sodium, potassium. There's other things like calcium, magnesium. Those would be ions as well. And then we also have other molecules within the cytoplasm or the cytosol. Organelles, you could think of the term organelles as like mini organs. So organelles are like the mini organs in a cell, just like your liver and your heart and your lungs for your body. So think organelles on the cellular level, these are the little things that help it function. So small complex structures that perform specific functions. So mitochondria would be an organelle, okay? Rough endoplasmic reticulum would be an organelle. And what else do I want to tell you? Inclusions, I briefly touched on last time, are clusters of a single type of molecule. So you could think of this, so for the example of melanin, that would be in your skin cells, but it's also in your hair. It's also your eye. That's melanin. Melanocytes make melanin. Melanin is the pigment that gives you your hair color, your eye color, your skin tone. Okay, so some of us have more skin tone in some areas, like freckles or something, right? So even though I'm fair skin, I have a gazillion freckles on me, so I have little deposits of that. And so within your cell, you'll have deposits of this as well. That's what an inclusion is. Also, glycogen would be considered an inclusion, and even triglycerides, because it's stored stuff in like concentrated area. That's an inclusion essentially. Various cells store glycogen, triglycerides, pigment. So now we're moving on to newer material, right? We didn't talk about this yet. This is new now. All right, plasma membrane. So now we kind of touched on it a little bit when we did the little brief talk about the cell last time. We're starting to talk about a little more in detail about the individual components of our cell now. So plasma membrane, also called a cell membrane, is responsible for the movement of materials in and out of a cell. So it says, I will allow you in or I will not allow you in. I will allow you to seep out of me or not. Okay, I'm in control of this. I decide whether you come in or out of my cell. If something's too large, it's not going to pass through that membrane. Okay, and it's a selective membrane. Some things it will allow to diffuse across its membrane, and some things it absolutely does not. You're going to need a protein to help you do it. So we're going to learn about that. And this creates the outer border of a cell, which you guys could see under the microscope or in the pictures of histology. And it separates the two main fluid compartments that we already mentioned. Intracellular, which is fluid within the cell, right? Versus extracellular, fluid outside the cell. So intra within, extra out. So there's another specific name that can fall under the category of extracellular fluid. Has anybody heard the term interstitial fluid? Okay, so interstitial fluid is also extracellular fluid, but not all extracellular fluid is interstitial fluid. It's like one of those math problems that you hate when they say all of A is equal to B, but not all of B is actually equal to A, right? So what we're saying is interstitial fluid is a more specific name for some extracellular fluid. And this is interstitial fluid is the fluid in between the cells. We'll see that more later this semester. You'll also definitely talk about it in 228, okay? Now we're going to talk a little bit about what's called the fluid mosaic model. So this is talking about the actual membrane, the plasma membrane, and what comprises it and what it allows in or out essentially. So remember before we talked about how the phospholipid bilayer, you had your polar heads, right? And they had their fatty acid chains or tails, okay, on the inside. And why do we have the polar heads facing the way that I did? Because we have extracellular fluid, which is a lot of water. That's going to be on the polar end. So here's your extracellular side, outside of the cell. So I'll put out. And here's inside of your cell. And this is where you have intracellular fluid because it's within. And so it also has lots of water in it. That's why your polar heads are facing the water side. And the hydrophobic tails, phobic meaning fear of, I don't want to be near, hydro is water, don't let me near the water, is on the inside of the membrane of this phospholipid bilayer. Other things that we have within this is the cholesterol molecule, which they put one here. And I'm sorry it's not like a blown up picture, but it's like a yellow squiggly molecule that they're showing there. What did you guys know or remember about cholesterol? Lipid-based, right? Steroid hormone, right? It also helps make our membrane stay more fluid. So it prevents the membrane from being too rigid. If our membrane is too rigid, it won't be flexible and it won't allow things to move in and out so easily. So we need cholesterol in there. It's a membrane stabilizer because it allows it to stay movable, okay, which is the idea of that fluid mosaic, movable model of the phospholipid bilayer or plasma membrane. So we have cholesterol embedded in there. And then do you guys see these blue globs here? Okay. These are what? Proteins, okay? Couple different kinds of proteins. Some of the proteins don't go all the way through the plasma membrane. They just may sit on the top or the extracellular side. They may just sit on the intracellular side. You can have proteins within your cell as well. But the proteins that we're talking about on the fluid mosaic model is whether it's only on one side or the other, or does it go all the way through like this guy does right here, okay? He goes all the way through the membrane. So he is transmembrane is what we want to say on that one. We're going to talk about him in a second. And then also, if you guys can see, we have these little green molecules sticking off some of our proteins. And we also have some green molecules sticking off of the phospholipid polar head. Okay, those are carbohydrates or sugar. So you actually have sugar sticking off the outside of your cells. Sometimes they're cell markers. Sometimes they tell other cells that this is you, this cell belongs here. I am me, I am self, okay? And sometimes it says, you don't belong here. Hello, immune system, come attack, get rid of this crap. It doesn't belong here, it's not me. And then sometimes we run into things like autoimmune disorders where it thinks it doesn't belong there when it actually should belong there. It became sensitized against itself. We don't really get to talk about that too much until you get more into 228. But if it's attached to a phospholipid, that sugar or carbohydrate, it's called a glycolipid. If it's attached to the protein, it's called a glycoprotein. So glyco means glycogen or think carbohydrate, carb, okay? So it's kind of telling you the word. Glycolipid, carbohydrate attached to a lipid molecule, or glycoprotein. Let's see. So coming back to our picture here. Another thing that you can think about, so if we're talking about the membrane structure that we saw here, we said that's a phospholipid bilayer. The phospholipid, you guys remember, kind of looks like a little bit of a bobby pin, sometimes with a crooked leg, sometimes not. Remember that crooked leg kind of meant that there is a double bond, so it would create a kink. The more kinks you have, the more fluid your membrane would be also. It wouldn't pack as tight. So that's why you don't want all saturated fats making up your membrane. You want to have unsaturated fats in your diet to help make a more fluid mosaic model, right? And we talked about the polar head, which was this guy right there. And then we also have the fatty acid tails, which is that end. The polar head will be hydrophilic. Philic means loving, hydro, water, water loving, right? So hydrophilic is the polar head. Polar made sense, right? That it would be attracted to water because it has partial charges, right? Polar covalent bonds. Fatty acid tails, though, are hydrophobic. They're not literally afraid of water, but they don't like it. So, and easy way to remember it is if, let's say, you were cooking or you have grease or something on your hands, working on a vehicle or fixing a door, I don't know, whatever, or cooking bread. So you have butter on your hands or grease on your hands. It's an oil-based lipid, right? So if you just stick your hands under water, the water doesn't get it off. It just beads up and it doesn't get it off. So you have to use soap. That ends up actually creating an emulsion and a micelle, so it can trap the fat molecules and allow them, when you put your hand under water, for it to rinse off. So just think of it that way. Hydrophobic, fat, hydrophilic, it likes water. So cholesterol, we already said, was stabilizing the membrane, right? So it helps provide fluidity to the plasma membrane. Glycolipid was something I already pointed out on the picture a second ago, helps form the unique sugar coating of our cells, because glyco, carbohydrate, lipid, it's saying it's attached to the lipid. And what else do I want to tell you? So it's kind of like a sugar fingerprint on the outside of your cell if you're into forensics, right? And actually, when I was reading in your textbook, it says that it's kind of like fingerprints, even like with identical twins, where it'd be very similar to an identical twin, but even siblings won't have identical sugar fingerprints on their cells, right? They might have similar, but it's not going to be the same. So I thought that was kind of interesting, but identical twins would have essentially almost identical sugar fingerprint on their cells. That was kind of cool. Let's see. So glyco for sugar, for glycogen that's attached to the lipid. Carbs like water. That's why it's hanging out in the extracellular fluid out there. Let's see. The diagram they want us to show, kind of also showing the fluid mosaic model. So basically, if I had a jar and I filled it with some water, and then I pour my phospholipids in there, what would be near the water side? The polar head, right? So here's my polar heads. But which way would my hydrophobic fatty acid chain tails go? They'd end up sticking up because they don't want to be near the water, right? Now, if I poured more water onto it, and let's say, oh, let's cover this up, then my phospholipids would kind of end up bunching up on top of itself, just like on our fluid mosaic model of our plasma membrane. And I would have more heads here, and my water that I poured on top would be there, and I would be preventing water from being in this region that is hydrophobic. Now, if I go back up to this picture that we saw a second ago, and you look at the protein molecule, I know it looks like just a blue glob, but what do you guys remember about what a protein looks like? First, it was just the primary structure, right? So you had just the sequence. Then it started to get like alpha helices or beta pleated sheets and hydrogen bonds to put them in that shape. And then from there, you had like a tertiary structure where you might have, some beta pleated sheets and some alpha helices. And if it ended up being a quaternary structure, you would have another protein or polypeptide bonding and attaching them together with hydrogen bonds and covalent bonds and so on and so forth. So this protein here is really just something all bunched up on itself, right? So this is going to have, you know, alpha helices, some beta pleated sheets, alpha helices, beta pleated sheets. That's really what that looks like. It's easier for us to understand like a solid structure, right? But what I want you guys to kind of understand about these proteins that are embedded within your phospholipid bilayer, think about where a hydrophobic area of the protein would be and a hydrophilic area of the protein would be. So the amino acids that are making up and getting linked together to make this initial strand some are going to be hydrophilic and hydrophobic, depending on what its structure is. And depending on how it bonds together, the hydrophilic parts that are polar allow for those hydrogen bonds. So all of this is like the same story, it's just saying it a couple different ways. And then if I stick it in my membrane here, the hydrophobic portions would be in the hydrophobic area of the membrane, and the hydrophilic portion of a protein would be on the hydrophilic ends. And then if you have a protein like this one, can you guys see there's a channel or an opening in the center of the protein here? So for example, these areas out here would be hydrophobic parts of the protein, and the area in here would be hydrophilic to allow a hydrophilic molecule to pass through. Because otherwise, hydrophilic molecules can't get in here. They'd get stopped, right? So I can have something called an aquaporin, which is a special protein that allows water to pass through the membrane. So you'll see that in a second. I'm just throwing it out now, like peppering it in there. You'll see it again in a second. But I want you guys to keep that in mind on why we're going to talk about proteins in a second and why it's allowing things to come through or not, whether it takes energy to do it or not. So that's what we're going with on this. So now if we go on to, oh look, membrane proteins. It makes up almost half the weight of the membrane itself. So that's quite a bit. So it's mainly phospholipids, but it also has proteins in it. And you have a little bit of cholesterol and you have a little bit of glycogen molecules sticking up off the tops on the extracellular side. But half of the weight of the membrane is coming from the proteins themselves. The proteins float in a sea of phospholipids. That's exactly what that picture looked like a second ago. And an integral protein is embedded in the bilayer. That's the transmembrane part. So if I come back up here, this guy here is an integral protein. He's crossing the whole membrane, okay? Versus this guy over here is not going to have that same name. So he is an integral protein or a transmembrane protein. Trans because he's crossing the whole thing. If we go back down, and then it says it can form channels for transport. Oh, look, it did. It formed a channel for transport in the picture. Cool, that makes sense, right? It can also create anchor points, so mechanical anchor points, so things can attach to those glycogen molecules that were hanging off of the protein. The protein can attach to something else also, like another cell potentially. So it also says cell-to-cell recognition. So that's part of our cell fingerprint, where I was saying, does it know that you belong there? Is this your cell, or is this an invading bacterium or something else? It can create, like I said a second ago, cell-to-cell connections. So that's cells holding on to another cell, so they'll link up by the proteins. And what else do I want to say? Enzymes. Proteins are enzymes, right? So we can have one of those proteins in the membrane will break something down or allow another reaction to occur, so that way we can do some other functions for our cell. I promise someday my whole voice is going to come back. I've never had my voice not come back, and it's like going on 3 weeks now. This is ridiculous. Don't get sick, guys. All right, so the other kind of protein, so I said we had integral, which was transmembrane across the whole membrane, phospholipid bilayer, and then we can have peripheral. Well, that makes sense. Peripheral, just loosely attached to an inner or outer surface of the cell. So it does not cross the whole membrane. That's the other guy that I said was just kind of sitting at the top there. So that's its function of a peripheral protein would be it could do cell-to-cell recognition. Do you belong here or not? And it also has enzyme properties that it can do. So this would be a peripheral protein that didn't go all the way through. It's just hanging off on the top, hanging out, has some sort of function, right? So if we look at structure of cell external surface, here I'll put this here so you can read both the integral protein. So this is one of those electron micrograph pictures I was telling you. Can you see how grainy it is? It's pretty much black and white. People are trying to do in research where they attach color to certain things. You guys don't have to worry about that now, but they think like certain things might have certain colors to it. But that's hard to say. It's not like looking at somebody's pancreas and going, oh yeah, it's this color inside of your body. We're talking about little tiny little things that doesn't really have light bouncing off of it to tell us that it is this certain color. But anyway, so it's kind of grainy looking. That's what it's talking about. So structures of cells' external surface. So these are things on the external surface of a cell. We talked about on that cartoon picture that you could have microvilli. And what did we say microvilli would do for a cell? They're like finger-like extensions that end up increasing the surface area. So for example, if I have my finger-like projection sticking out, more things could pass in between my fingers, on the tips of my fingers, in between. If I chopped off my fingers, do I have as much surface area? No. So that's why think about the fingers. You can get more area of the membrane and it doesn't take up as much space. Think of it's kind of compacted. So right here is like a whole bunch of little finger-like microvilli projections. Okay, like that. So on and so forth. And not just increasing surface area, but that allows you to have an increased ability to absorb things. So cells, for example, that are in our GI tract that need to absorb things will tend to have a lot of microvilli on it. So if you want to see microvilli, find something that's in the intestinal tract, intestinal lumen area, so that you can find that structure. I'm not likely to have these microvilli, say, on my squamous epithelial cell, right? So if we look at, let's see, so microvilli also means there's more plasma membrane. It's important in cells that have absorption functions. We find these cells with microvilli in cells lining the intestinal tract, mainly the small intestine where you need to do that. The term lumen means like the opening through a tube, let's say, right? So the intestinal tract has a lumen through it where food or waste products are now passing through, right? And so it's a lumen just means an area inside an area. So it could be inside a blood vessel would be a lumen where the blood, red blood cells, white blood cells are flowing through in the plasma. That would be through the lumen of the blood vessel. We can also have things called membrane junctions, which are connecting and supporting cells. And there's three that we're going to talk about here on membrane junctions. We have tight junctions, we have desmosomes, and we have gap junctions. So membrane junctions basically help you form tissues because if it's connecting another cell to another cell, now you start having multiple cells linked together, you now have a tissue, right? So connect and support cells, it's found on the external surface. And of the 4 tissue types that you guys, did you have to know this last week or starting this week? 4 tissue types. Epithelial, connective, muscle, and nervous, right? Your 4 main tissue types. Of those 4 types, which one needs something to hold them together? What do you guys think? Epithelial, and also you'll find out muscle. Okay, epithelium, muscle for sure need things linking them together to give it stability, right? Otherwise, you could like pass something through your arm. And anybody see Valerian and the cool like alien creature that could morph into anything else, and she could like morph her body and like the knife went through her, but it didn't hurt her until the end. It did hurt her, but anyway, think of it like that, right? You need stuff to hold it together. Otherwise, you'd be very weird fluid-like. I don't know. Okay. If you haven't seen the movie, it was kind of a good movie. Check it out. So membrane junctions connect and support our cells. There are 3 types that you guys need to know about that I want you to know. So tight junctions are rows of integral proteins that spot weld adjacent cells together. So spot weld would be like if you had two pieces of metal and you put another piece of metal and you melt it together so it sticks the two pieces together. That's a spot weld, right? You could think of it like snaps on a shirt. So if you have a good tight snap, it snaps it together. It's not going to let something really pass between it. There's not a lot of space there, right? So you can think of it like snaps or spot welds. And so it's close to the tissue surface. So this here is the apical surface or the outside surface of the cell. This here would be the basilar surface or basement membrane area of the cell. Apical surface means like it's to the opening of like the lumen or the outside of your skin to the outside world. So a tight junction would be near the surface, the apical surface area. So close to tissue surface, it prevents unwanted material from leaking into the body. So it's a tight junction. It's not letting stuff through. Okay. So again, spot weld or snap that's holding the cell together. Then you can have something called a desmosome. They're short linker proteins that cross the space between cells to hold one to another. So a good example is like skin, and a desmosome looks like kind of like shoelaces, right? And on shoelaces, you could adjust the tension between it. So you could make it a tighter, tie between the shoelaces, or you could loosen it and have it a little loose. So that's the idea of this too. You could change the tension between the desmosome proteins to allow a closer fit between the cells or a little bit more space between them. And let's see, so with most commonly seen with skin cells, so for example, stratified squamous epithelium. If you guys look at the layers of the skin, when you're trying to find like stratum this, stratum that, stratum spinosum is like an artifact of this. So it would be kind of like if you peeled it apart at the stratum spinosum layer, you would see like these little mini pokey things sticking up. That would be the desmosomes holding that area together. So, so for example, with water, okay, so stick your hand, your epithelial cells, in a thing of water. What if you do, like you hang out, you go swimming for a while, you go in the lake or you're in the bathtub? What happens to the cells of your skin? Water is allowed to seep in, right, over time, not just like for 5 seconds, but if you're hanging out, you're like a fish out there for a while, you're hanging out, the water gets in and the skin is allowed to expand because the desmosomes allow that expansion between the cells. And then when you get out of the water and you allow the water to kind of like evaporate off eventually and seep back out, then you'll go back to looking normal again, right? But you'll kind of look a little funky in the interim while you have that excess water in there. So the desmosomes allow that water to come in or come out, but not too much, but enough that you see a difference, like with your fingers, you get all pruny looking and stuff, right? So that's desmosomes allow that to happen. And then the third one that we have is gap junctions. So the gap junctions are this last picture here. It's communicating junctions between cells. They're made by hollow protein tubes called connexons. So the connexon is a protein that is like a hollow tube, and it's allowing stuff from one cell to pass to the cell on the other side of the connexon. So there are times, for example, where you could get an injury, you don't know it, meaning your brain isn't aware of it yet, but already your cells are communicating with each other and doing something about it before your brain even gets involved. And it's because of these connections where it's transmitting like, oh my gosh, I've been injured, help, help, help. The other cell can't really help you, but at least it knows about it, and maybe it can bring histamine or other things to wall it off in that area so it swells up in that local area, and the brain didn't even know it yet. Kind of like when you maybe got stung by a mosquito. You don't feel it immediately. You start to feel it afterwards when all the histamine is there and you're getting itchy and you're like, it bit me. Stupid thing, where is it? I can't even smash it. It's already gone, right? Your body was already making that talking connection between other cells locally. And this is why. It's because gap junctions allow that. What else do I want to tell you? Also, so you can think of like a gap junction. Don't think of it like a gap, like a big space, but more like a bridge between the cells. Think of it like a bridge. So it's more like bridging the gap between cells is a gap junction, I guess. So this allows substances to pass between our cells. A good example would be like heart cells, okay? or other like smooth muscle cells or something like that. But heart cells, it would allow like an electrical impulse from one cell to another. So it could be electrical, it could be something in the cytoplasm, like a chemical reaction going back and forth, telling the other cell, oh my gosh, we're being invaded, help, 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 you know, that kind of thing. So that's what you want to think about when you're talking about. So a connexon, now that you know a little bit about proteins, is it a peripheral protein or an integral protein? It has to be an integral protein, right? Because it's going through the membrane and allowing something to pass from the inside of one cell to the inside of another. So a connexon would be an integral protein, an example of that. Or transmembrane protein, if you want to say transmembrane. So that's our gap junctions there. So allows substances to pass between our cells. An example would be like heart muscle cells. Little m is my abbreviation for muscle. Heart, symbol heart, you get that one. All right, so membrane transport. This is all about how things are allowed to move in or out of the cell. So, for example, a definition, we have interstitial fluid, we said was fluid in between cells. Interstitial fluid is an example of extracellular fluid, right? But remember, not all extracellular fluid is interstitial fluid, but all interstitial fluid is a just more specific name for a type of extracellular fluid, okay? And let's see, what do I want to tell you about it? So I answered the question. I was going to ask you, is interstitial fluid extracellular? I was going to ask you that. I gave you the answer. All right. Let's see. How about the term semi-permeable? So semi-permeable, semi means kind of, right? Semi-permeable, permeable means allowing something to come in or out. So we're saying if something is a semi-permeable membrane, it means only select substances are allowed to pass through. It's selective. So not everything is allowed to pass when something is semi-permeable. If it's permeable, it means anything could pass. If it's impermeable, it means nothing can pass. But our membranes are semi-permeable, so we allow certain things in and we don't allow other things in. We're selective. And let's see. We're going to look at now passive process versus active process. So we have passive is number 2 here, and if I scroll down for a second, then we'll end up talking about active process at number 3. So let me go back up to passive. Hope I'm not making anybody dizzy. All right. So passive process means it does not require cell-generated energy. Instead, it only uses kinetic molecular energy. So what that means is kinetic means movement, right? So we're saying molecules move around. Like you guys know, we talked about how electrons can bounce around super fast. Molecules have a little bit of a vibration to them and they can move around. If you see like one of those nature shows on TV and they look at the cell of something and they show you how everything is like moving on its own and you're like, ooh, how's that protein know how to move from here to here? Or I mentioned before on the cartoon picture of the cell that it can move along things like tracks within the cell and it knows where to go. So in this sense, we're just talking about like small particles, not as small as an electron, larger than that, but molecules that are able to move. Okay, and so if we say that it doesn't require cell-generated energy, passive means it doesn't need ATP. Okay, but it's using its own method of where the molecules want to flow to generate some energy. Okay, or charge in a little bit. So passive process. So if anybody's familiar with like physical therapy, for example. Or if a chiropractor wants to see if you do you have a problem with active movement versus passive movement? Active movement means you had to physically lift your arm in the range of motion to see if you could do it. Passive movement would be, hey you, let go, relax your arm, I'm going to move it for you. Let me see what kind of range of motion you have. I do the work for them. I'm going to move their arm. They're being passive. They're letting me do it to them. They're not actively doing it. You can think along those terms for this. So passive, we have diffusion, which is just movement of a substance down its concentration gradient. So what that means is, if you have a concentration of something, like they're kind of showing you in this little beaker picture here, and you do nothing to it, you don't stir it, you don't mix it, you do nothing. You just put whatever your substance is, whether it's liquid or solid, I don't care what it is, but you put it in the center, you do nothing else to it, all by itself, it will diffuse out because it doesn't want to be where it's super concentrated. It has all this other space in the beaker that it could spread out to, okay? So what it means is where it's high concentration, it wants to spread out and go where there is lower concentration. Eventually, if you didn't stir this beaker up at all, it would eventually reach a homogeneous or homogeneous solution. It would be equal throughout. So they're showing you like red dye particles that they put in the beaker of water here. And if they don't stir it up, they don't jiggle it, they do nothing to it, no heat, nothing. It just put the dye in, leave it alone. It starts to spread out from high concentration, which was in the center where there was a bunch of those red dye molecules, to low concentration where there was barely any of it. And eventually, it's going to be equal throughout, no matter where you sampled it. Homogeneous, right? And that's, you didn't do anything. That's its natural kinetic movement of the molecule itself is going to do that. It's always going to want to go from high concentration to low concentration. It wants to spread out. So you can think of it as if we all went, this is like the club in here, right? And we're all in the middle of the dance floor. At some point, I mean, you might want to rub up against other people, I don't know, but at some point you might want a little space. So you're going to spread out and use the whole dance floor, right? And not just be all, everybody's like here when you have this big dance floor that you could be on. So think of it like that. You want to spread out so you're not banging into everybody every 2 seconds, unless you're somewhat of a perv. But anyway, or not, you're just horny. I don't know. Anyway, moving on. Okay, let's use another analogy, right? So simple diffusion is just small substances move directly through the bilayer, if we're talking about like the phospholipid bilayer, right? So what that means is nonpolar molecules, okay, and we know lipids, for example, are a nonpolar molecule. They would be able to pass through this membrane, okay, because it's nonpolar on the inside, right? And if we looked at, for example, oxygen and CO2, they are nonpolar molecules, right? So if we drew out the structure for O2, it was this guy here, O double bond O. It shares the electrons equally. That's why it's nonpolar. It's not a polar covalent bond. It's a nonpolar covalent bond. So it's just nonpolar. It wouldn't want to interact with water, right? Because water is polar. O2 by itself is not polar. CO2 is not polar. So I drew its structural formula too. It's sharing all their electrons equally, so it's nonpolar. So what it's saying here, interstitial fluid, is that extra or intracellular? Extra, so this means this is the outside of the cell here, right? Because it said interstitial. Cytosol tells you it's where? Inside the cell, right? Or intracellular fluid. And they're showing you the oxygen molecules are these red dots. And where do we have a higher concentration of those oxygen molecules? On the outside of the cell. And where do we have a lower concentration? Inside of the cell. Where will oxygen want to move if it's on the dance floor? It wants to go from an area of high concentration where there's too many of them to an area of low where it can spread out. So it's going to go into the cell. And because it's nonpolar, it can pass through the membrane. It doesn't need anybody to help it do it. We could go outside the club through the membrane doors. That's right. I like that. Let's talk about inside and outside the club. There's a whole nother, so this is where you have like the dance off out in the street, right? So they have to take it outside. Are you guys following me? All right. If we look at carbon dioxide on the right-hand side of the picture, where is there a higher concentration of the carbon dioxide molecules? In the club, right? Inside the cell, the cytosol, and lower concentration in the interstitial fluid or outside of the cell. So which direction are the carbon dioxide molecules going to flow? From high concentration to low concentration, meaning from in the cell to out the cell. It's going to go do the dance off outside. Okay? I like that. Yes. Hey. I can't dance like those people in the dance off though. So somebody else is going to have to like do the break dancing and the robot and the other stuff that, what's it called? Dubstep and all that other stuff where they can, it's amazing how people can move their fluid mosaic model. Those fluid bodies when they do all that kind of crazy, I don't know. Anyway, moving on. Let's see. Okay, so I talked about that. So simple diffusion means you don't need any help. It's just going to go because of its concentration gradient. These particles of oxygen or CO2, you could think of them as particles of other things as well. In this case, we're talking about these nonpolar molecules that can pass through only based on its concentration gradient. That's it, okay? Now, sometimes you need somebody to help you get across, and that's what's called facilitated diffusion. You need a facilitator. So this is like somebody at the door of the dance club. You give them your ticket, and they say, I will let you in. I'm going to usher you in, and I'll walk you into the dance floor, or you are being naughty, and I'm taking you outside, right? You're not allowed in the club anymore. So you need a facilitator. A facilitator in this case is a protein, okay? Now the protein can just have a channel that allows, for example, in this picture they're using sodium. Sodium is polar or nonpolar? It's polar. So it can't just go through the membrane by itself. It can't do simple diffusion. It needs a facilitator. So facilitated diffusion, it's using an integral protein that has a channel. The channel itself is hydrophilic, which will allow the sodium molecule to pass through, versus the hydrophobic areas of the protein that's allowing it to be embedded in the hydrophobic part of the membrane or the doorway of the club, right? So, but sodium is still going to go down its concentration gradient. So where do we have more sodium? Outside the cell. Where do we have less sodium? Inside the cell in this example. So it's going to go from outside the cell to inside of the cell, but it needed a facilitator. That's why it's called facilitated diffusion. No energy is needed to do this. It just needed a helper protein to allow it in. If you look at potassium, where do we have more potassium on this picture? We have it inside the cell. So where does potassium want to go? Outside where there was less. High concentration to low concentration. Did I mention before in one of the other lectures about sodium or potassium leak channels? Did I say that yet? So sometimes they have, this is an example of those leak channels. There is a protein that is allowing sodium or potassium to leak in or out. Did we say where we tend to have more sodium or more potassium? Typical of a cell. We typically have sodium on the outside of the cell and potassium on the inside of the cell in higher concentrations, okay? So we typically have more potassium inside, more sodium outside. So these leak channels are allowing sodium to leak in where there's less sodium, and allowing a separate protein is allowing potassium to leak out in this leak channel, okay? This will be important in a little bit. That's why I'm mentioning it now. The picture next to it is another carrier protein. So we talked about ions would need to have facilitated diffusion because they're polar. Glucose tends to be somewhat of a large molecule, and so it also needs help getting into the cell. So it will also use a protein to allow it in or out. Usually we're trying to bring glucose in, right? And the interesting thing about this protein is the carrier protein will actually change its shape. So again, this protein, think of it like an enzyme, right? It's specific for one thing. It is only allowing, in the first picture, sodium through on the first protein. It's only allowing potassium through on the other one. A potassium leak channel is not going to allow sodium through and vice versa. A glucose channel is not going to allow any other shaped molecule in other than glucose. It's very specific, okay? So what it does is it's open on, in this case, the outside of the cell. Glucose comes in and touches and it goes, oh yes, you fit in here. I will allow you in. It then changes shape and then, I gotta make a spit sound, allows glucose into the cell, right? And then it will change shape again and open back up to the outside of the cell ready for the next glucose molecule. Anything other than glucose touches it, it doesn't do anything. But when glucose fits, the puzzle pieces match, it allows glucose in, changes shape, sticks it on the inside, changes shape, opens back up to the outside. That's what it's doing. Does not take ATP to do this. This is just facilitated diffusion. You needed a helper person, a protein in this case, to allow you in or out of the dance club, right? Okay, nice. And then we need something like water, right? So we could have, where remember I said we have a specific protein just for water called an aquaporin. So aqua for like agua, agua fria, cold water, 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 right? Agua, Latin, aqua. So aquaporin, a pore, something allowing something to pass through, and it's going to allow water to come in or out of the cell, usually into the cell in larger, faster amounts, okay? We can decide how We can decide if we need to add more water into our cell, we can put more aquaporin proteins into our membrane. Maybe we don't want any more water coming into the cell. We're going to take that protein out. So it's not going to let so much pass in. Part of the fluid mosaic model. That membrane, that phospholipid bilayer is changing constantly. So we can put proteins in to allow us to get more fluid into the cell. We're getting a little dehydrated. We can take it back out. We can move things around. It's always moving around. Those phospholipid heads, you guys, are not attached to each other. They're not covalently bound. It's just hydrophilic and hydrophobic properties that keep it together. That's it. That's why I think it's like so fascinating if we really were like that creature thing. You kind of are on your membrane, right? In Valerian? Yeah. Okay. See, if you haven't seen the movie, you have to go check it out now. Go rent it for free at the library or something, but check it out so you know what I'm even talking about. So aquaporins, facilitated diffusion, does not take any ATP or energy to do it, but water is also going to flow with its concentration gradient, okay? So where you have high amounts of water, it's going to want to go where? To the low amount of water, okay? So in this case, you know, you don't really think of water as being a particle. You don't think of it being solid, but it's a molecule. Particles of water are deciding whether to go, and even with the aquaporin, with facilitated diffusion, it's still going to go from an area of high concentration of water to an area of low concentration, which brings us to a more specific term called osmosis. This is a special case. It's simple diffusion. This does not need the facilitated protein to help it. This is kind of led into water, but this is talking about still water-only movement based on its concentration gradient. So where you have high concentration of water, it's going to want to go where there's less water. Another way to think of it, okay, is if you had other particles of something else, which we'll call a solute, right? Other particles, some sort of solidish other molecule. If you have a lot of solute somewhere, can you have a lot of water there? No. So if you had this solid particle here, right, a bunch of them together making a mass, you might have a lot of water around it. Where will water want to go? Where there is less water, meaning where the solid particle was. So it's trying to get in between there, and eventually it would become homogeneous or homogeneous, just like that beaker did with the dye. So what it's trying to show you in this picture here of the U-tubes, okay, not YouTube, but it has, the important thing here is there is a semi-permeable membrane that is splitting side A from side B, okay? It might be a little bit hard to see, but the red dots are actually like the little Mickey Mouse heads, which is the water molecules, so the oxygen with the 2 hydrogens on it. So if, they did the pictures wrong, just so you know, there's some typos in here. But technically, right now, in this first picture, number 1, they're saying there is equal amounts of water on either side. So you should be able to count all the water particles on one side of the semi-permeable membrane to the other side, and they should have the same number, 8 and 8, or 10 and 10, or whatever. And the solute, they designated as the dark gray or blackish dot, right? So if it's equal, it should be the same on either side. Now the semi-permeable membrane only allows water to pass through, not solute, okay? So if it's equal on both sides of solute and only water is allowed to pass through, if you didn't do anything else, no energy is needed, you would eventually at some point have equal amounts of water on either side, okay? What they're trying to show you is, let's say though, you have more solute on side A. So we have 1, 2, 3, 4, 5, 6, 7, 8 solutes on side A. We have our semi-permeable membrane. And we only have 1, 2, 3, 4, 5 solutes on that side. What did I, 1, 2, 3, 4, 5, 6, 7, 8. I said there was 8 on this side, right? So which way will water want to flow? Because only water can pass through this semi-permeable membrane, not the solute. Which way will water go? To side A, because there's more solute there. It's more concentrated as solute, meaning there was less water there. So water is going to flow over there. It goes down its concentration gradient from high water to low water till it would reach an equilibrium. And when I say equilibrium, I'm not meaning equal heights of the water until it's diluted that other side to the equal parts of the side B, okay? So that's why it's showing, I think I think 1, 2, 3, 4. So this is where the typo was. They only had 4 on this one and 1, 2, 3, 4, 5, 6, 7, 8 on this side. They should have had only 4 on this side also because I said the solute doesn't cross the membrane. So you can cross off one of those black balls because it should have been equal to the first picture. So don't get confused. Solute doesn't cross the membrane, only water does. Okay, that's what I want to make sure you guys understand. If you're trying to look back at this later and you're like, she lied. No, they did it wrong. They drew their picture wrong. So with this, what do I want to tell you? Okay. High and water, we did all of that. Very good. So now I'm going to draw a picture to describe what osmotic pressure is, or an osmolarity meter. Osmolarity is usually talking about like the saltiness of something. Ions are salts. Okay, so like sodium, you could see how much particles of sodium in the water was and test that. So essentially, if I draw a beaker here, okay, and I have a semi-permeable membrane there, which means only water can pass through it, nothing else. We'll call this side A, we'll call this side B. And on my thing, though, of the water, I have a lid on side A, so it can't move up or down like we allowed it to in the YouTube picture up above. And then it has a meter on it that can measure 0, 5, 10 for 10 osmolarity, or milliosmoles is usually what you'll see it listed as. And so if I put, say, 5 particles of sodium or something in here, so 1, 2, 3, 4, 5, right? The other side doesn't have it. Solute can't move either way. Which way will water want to flow? From what to what? It's going to want to go from high concentration of water to low concentration of water. And if we have solute taking up space, it means there's less water there. Or you could think of water wants to dilute the solute. So water is going to flow across the semi-permeable membrane from side B to side A. But the problem is we have a lid on that side. So we're not allowing the water level to change, right? It can't move. But it's going to exert a pressure against that osmolarity meter. And that's how it's measuring osmolarity, okay? We talk about these pressures a little bit later, and that's why I'm bringing it up now. But this really has everything to do with tonicity. So let me see what else I want to tell you. So because the fluid level can't raise up, you saw which way it goes. It's exerting, so water is exerting a pressure onto that side, even though it can't move and it's not allowing things to do anything else. So that is that. And then I want to ask you, what am I going to ask you? I'm going to say, where does water want to go? I already asked you that. See, I'm ahead of myself. Darn it. But then there's the piston on top. Water can't move. So what would happen to the pressure reading if you doubled the number of particles? Now let's say we have 10. Now we have 10 particles there. What would the meter read now if we double the amount of particles? It would double the amount of pressure. Okay, that's what I'm trying to get at with that. So, and what would happen if you made the membrane permeable now to the particles? Let's say the membrane now allows sodium to pass through it. Where is sodium going to go? Then sodium would want to go, it could cross the membrane because we're saying we're allowing it now. It would go to the other side, and water in it would end up coming to an equilibrium at some point, right? Eventually. So now we're going to talk about tonicity, and I need to speed it up. So tonicity, the ability of a solution to change the shape of a cell by osmosis. So it's talking about where is water flowing in relationship to where we have all this higher concentration of solute, okay? So isotonic, iso means the same. So isotonic means there's the same number of particles on both sides of a membrane. Hypertonic would mean what to you? More particles. Hypotonic, hypo means less particles. Okay, so I drew 3 beakers here, and I filled it with some water, and I put 1 cell in it, okay? Now in this cell, let's say on the first picture, I have only solute inside the cell, okay? Let's say on picture number 2, I have only solute outside of the cell. All right? And what would happen in picture number 1? Okay, which way would water move? Into the cell. So I'm going to put water is going to move into the cell. It's going to end up diluting that cell. The terms hyper and hypotonic are relative to what you're talking about. So for example, who would be hypertonic in this picture? In number 1. The cell is hypertonic because it has many more particles inside of it, meaning that the water is hypotonic that was outside of the cell. So think of it as a relationship term. In number 2, which way is water going to go? It's going to leave the cell because there's all these particles outside of it, not in it, right? So water would end up leaving the cell. Who is hypertonic? The solution outside of the cell. That's a good way of saying it. Yeah, the solution outside of the cell is hypertonic. And who is hypotonic? The cell is, because it didn't have any particles in it, or less. If I say 1, 2, 3 particles and 1, 2, 3 particles, what does that mean? It's isotonic. It's equal inside of the cell as outside of the cell with the number of particles. Which way would water flow? It actually is, on all of these pictures, water is going to flow in and out of the cells on both of them. It's just, in the first picture, more water is flowing in than is actually coming out. In the second picture, more water is flowing out than is coming in. And in this third picture, the isotonic picture, it's actually equal amounts are coming in and out of the cell. So the cell isn't going to get ballooned up or shrink down for any reason if it's isotonic. So if we look at the picture of the red blood cell examples here, okay, so the first picture, if it says no net movement of water, is an example of what? Isotonic. The middle picture says water is entering the cell. So if water is going in, it means the cell is hypertonic, right? Water's flowing into the cell because there was more solute inside of the cell. And then the last picture, the cell is, because water's leaving it, the cell is shrinking, it's losing its water. So it must mean the cell is hypotonic. I'm going to put cell is because I think you guys are still slightly confused. You have to decide if you're talking about the cell or the solution around it. It's a relative term. Look at where the number of particles are and which way water is flowing. Water is flowing due to the osmosis stuff that we told you about. These are semi-permeable membranes because they're cells. So go back and review that. It'll start to make more sense. Real quick, it says filtration. So we're still talking about diffusion. This is another passive process, is filtration. It's a pressure gradient, pushes the fluid across the membrane. So you could think of an example like a coffee filter when you're making coffee and you're pushing water through the filter. That's your semi-permeable membrane. And an example for us or humans or mammals, and we talk about this in 228 a lot, is blood pressure is this same mechanism. So high blood pressure would push fluid out of the blood vessel and into the surrounding tissue. And then that increase in the pressure that would be experienced in those areas is how we're determining what's going on with blood pressure and an increased pressure and the filtration of that. Okay, so we talk more about that in 228. Just know an example of filtration for us might be blood pressure. Okay. For an active process, this is actually requiring cellular energy. And it means that substances are moving from low to high concentration. Well, what does that mean? We're pushing it from an area where there isn't very many of it, and we're shoving it on the dance floor where everybody's already too crowded. That's what we're saying. That means this is the opposite of passive diffusion. Opposite, okay? And in order to do that, this is an active process. It's going to take energy to do it. So it means we either need ATP, our energy molecule, like in the form of making a pump work, because we're pumping it against the concentration gradient. We're going the opposite of what makes sense. Okay, so in order to do that, it's going to take work to shove more things in the wrong direction. or we would need a form of kinetic energy to allow this process to occur. Okay, now if we're saying primary active transport, it means that ATP is used. Okay, and if you hear the term primary active transport, it means you're going to need a pump to do it. So we have this thing called the sodium-potassium pump. Okay, and the sodium-potassium pump, basically, we said earlier that there were sodium and potassium ions and a certain concentration of each cation inside and outside of the cell, right? So we said we have this cell. We said that we have more what, where? More potassium is inside the cell. And we have more what outside of the cell? More sodium outside of the cell. Okay? And then I had mentioned before that we actually have those leak channels that kind of let sodium leak in because sodium is high outside, low inside. So it's going to go down its normal concentration gradient. And I said potassium will have some leak channels that allow it to sneak out of the cell where it was high concentration inside, low outside. So that's regular passive diffusion, right? When we talk about the leak channels. But if we let too much of this stuff leak out, our cells aren't going to function very well. So we need something to force it back to keep what we're going to learn in a second is called a resting membrane potential. So in order to say, whoa, potassium, you need to go back in, and I know you're still high on the inside, but I can't let too much of you leak out. This is like a leaky boat, and we're going to have to use a pump, a bilge pump, to prevent too much water leaking into our boat. But we're constantly allowing it to leak. So we have to pump it back opposite its concentration gradient and force it to do what we want to keep a resting membrane potential. So that's what this picture about the sodium-potassium pump is trying to tell you. What happens is you have a protein, which is this, in this case, a pink glob, okay? And what it does is it is going to take 3 sodium, okay? And it needs ATP, so you get the little starburst picture of ATP, means you need energy to do this. This is our sodium-potassium pump. Pump means we need ATP. Okay, so we have high concentration of sodium outside. So if we're going to go against our concentration gradient, which way are we pumping 3 sodiums? It's high outside. We have to go against the concentration gradient. So we have to pump sodium out. So we're going to take 3 sodium and spit it out with this protein. It needs the protein to do it. And we need ATP in order to do it because we're going against the concentration gradient, the opposite of what makes sense. We're going from an area of low to an area where it's already high. That's why we need energy to do it. And then the protein's going to change shape, and it's going to take 2 potassium and shove it in. Okay? Because it's going against its concentration gradient. So we're going to take 2 potassium and shove it in where it's already a high concentration of potassium. Okay? And then when we cleave off that phosphate of ATP and we break it down to ADP plus a phosphate, that's when it changes its conformation on the protein. We'll do more detail of this later. But that allows the protein to reset and do it again. So the sodium-potassium pump is our bilge pump. We've allowed, through regular passive diffusion, sodium to leak in. We're allowing potassium to leak out. But in order to keep our cell happy, we got to force it back to keep a high concentration of potassium inside, high concentration of sodium outside. That's why we have to have ATP to do it. We need a protein pump to do it. And we're going against our concentration gradient. We're putting it where it doesn't make sense, technically, if you're thinking of concentration gradients. The other idea is a secondary active transport, or it's also called coupled transport. So this moves substances against its concentration gradient by harnessing kinetic energy. So this is energy that is not ATP. So primary was ATP pump. Secondary means no ATP, but we're moving substances against its concentration gradient. So this is secondary active transport. So it actively means we needed energy. It's just not ATP energy. And there are 2 ways we can do this. We can use a symport or an antiport. Another name for symport is co-transport. Another name for antiport was counter-transport. So let me explain it, mull it over, look it over again later, okay? So basically we're saying that we are moving two things, two different substances. We're either moving them in the same direction, so think like together, or co-transport. They're going in the same direction. So that would be like me saying they're both going into the cell or they're both going out of the cell. Versus the antiporter, anti like opposite, against, or counter-transport, means that you have two substances, but they're going opposite directions. One's moving in while the other one's moving out. Okay? Now, the difference is, so a symporter co-transporter is moving substances in the same direction. So its protein is called a symporter. So it's a symporter protein that is allowing you to move them in the same direction. The antiporter is an antiporter protein, is what we're calling it, that is moving them in opposite directions. And so one substance is going down its normal concentration gradient from high to low. That makes sense, right? That's the energy, the kinetic energy that we are making to allow the other one to go against its concentration gradient. So big picture is we have one substance going with its concentration gradient from high to low like normal, like a passive diffusion would have, but the other substance that has to move is either it has to go against its concentration gradient. And either they're both going in the same direction, so it'd be a symporter doing it, they're both going into the cell, let's say, or it's an antiporter, and they have to go in opposite directions. One's moving in while the other one's moving out. But still, of the two substances, one is going down its natural concentration gradient and the other one is going against from low to high. I know it sounds weird. So draw a picture of it, okay? And I will talk about that when we come back on Tuesday. But let me zip through this last part. If you have to leave, just look at the end of the video before, because I want to get all your exam one material on here super fast. So I will come back and draw that on Tuesday, but I want you to try to draw what we just talked about there. So there's a picture of the antiporter to help you draw your picture. So use that to help. Vesicular or bulk transport. This is transport of large quantities of material. So it's still an active transport. It needs ATP to do it. So it's moving more large amounts of doing something. And the way we can do this is either through exocytosis or endocytosis. And remember, we already drew a picture of this and talked about that. So exocytosis, exo for exit the cell. So this is material inside a vesicle, like a little membrane-bound circle that's transporting things to the outside. So it's membrane-bound, it exits the cell, and it dumps the contents outside of the cell. It needs energy in the form of ATP in order to do this. Or the vesicular bulk transport could be bringing things into the cell. That's endocytosis. So material moves from out to in, okay, on the cell. So this is also inside of a vesicle, okay? They're kind of showing you a picture here. If you guys go back to the top of the notes, this is, no, don't do that. Remember when I drew these pictures? Endo and exocytosis, that's this. Again, draw it yourself. I'll redraw it for you later, but I just wanted to show you that real quick because you guys have seen this. I already talked about it. And really fast, phagocytosis is cell eating, and it would be like solid material, okay? So like let's say you have a bacteria that you engulf and you're going to break it down and eat it. Or you might have products of a cell. You know, maybe you have a mitochondria that's kind of not doing so good. It's starting to break apart. So you're going to chew it up and recycle the parts, essentially. So cell eating, solid material, is phagocytosis. You guys have probably heard phagocytes, where it's a whole cell engulfing something and bringing it in. Pinocytosis, I mentioned you guys before. Think Pinot Grigio, like wine. This is cell drinking. So it's liquid material being brought into the cell, essentially, in massive amounts. This is bulk transport, and you need ATP to do this, okay? Then I briefly mentioned resting membrane potential. So this is the cell membrane creates an electrochemical gradient. Cell membranes are responsible for maintaining the different concentrations of materials with a charge on either side of the membrane. So this is going back to me talking about sodium ions and potassium ions. Okay, so we're saying voltage is the separation of charged particles. This is going to come back, and we're going to talk about this when we talk about muscle physiology in the middle of the semester and nerve physiology at the end of the semester. How do we make a muscle cell contract? How do we make a nerve cell send its signal? This is where it's going to come from. It's going to come back to resting membrane potential. So the resting charge inside of the cell is different from the outside of the cell. So this big circle I drew here is just a general cell membrane, right? And inside of your cells, you will also have a bunch of negatively charged proteins that tend to be near the periphery of your cell membrane, okay? And we also have what I said, remember we said like you have the potassium leak channels. So I'm going to draw a couple of potassium leak channels. So that's just allowing potassium to leak out, right? Because we said we have a bunch of potassium inside our cell. It's positively charged, it's a cation. So it kind of leaks out a little bit, and there's less concentration outside. And then I said we also have sodium leak channels, right? So that's what the circles are going to be, the leak channels. So we know we have lots of sodium outside of our cell, and they're wanting to leak in, so we'll have like a little sodium inside, right? But the majority of it was outside because we said we have high concentrations out. And then we have what are called our sodium-potassium pumps. So this takes energy, right? Because we're going against our concentration gradient. They're all over the surface of our cell. Okay, so that's a highlighter, so I'm not going to draw it with pink because it'll look kind of funky. But basically what you're doing is you are pumping how many sodiums out of the cell? So we're doing 3 sodium pumped out for 2 potassium getting pumped in with our sodium potassium pump. I should have drew it with the pink dot there, right? I'll do that real quick. So pink, and it's one protein doing both of them at the same time, right? Because it has to do both. And so basically what I'm telling you guys here is that we have our blob of a cell. We've achieved a charge separation, right? Because we have negative charge on the periphery of the inner part of our cell. We have positive charges on either side of it. What you guys will realize later, especially with nerve cells, is if we were to measure what the polarity was inside of the cell, it would be negative 70 millivolts. That's an M. Pretend like that's an M. Negative 70 millivolts is what that would read out to, okay? So basically, to maintain the separation of charged particles because of those leak channels, we use that sodium-potassium pump to force it to stay high concentration potassium inside, high concentration of sodium outside. It maintains this resting membrane potential so that when it's time for our cell to do something, like the muscle cell or the nerve cell, when we decide to depolarize and do something, it won't be able to do it if we can't maintain this resting membrane potential. So keeping the charges separate on either side of the membrane is what allows us to do that. That's the resting membrane potential. So all of that, guys, from there up is exam 1. That's it. I'm sorry we went over. Exam 2 starts with Roman numeral 3 for cytoplasm. So everything from that we talked up to today is all exam 1 material. Use the, what's that thing called? Study guide. Sherry already turned on the learning pathways. You got to go through each step before it allows you to move to the next to finally see the exam at the end. It's trying to teach you how to study. So utilize that tool in the A&P website. Make sure, internet students, you sign up for your exam. You guys, on Tuesday, we will start exam 2 material and move forward. Thursday will be the exam in this room, 1 p.m. on Thursday. So 1 week from now, you have your exam. You guys should get into small study groups. You guys should be quizzing each other. You guys should be doing anything you can. Have your notes in front of you while you do all of that stuff. I'm going to put the clicker quiz on now. I'm sorry we went over. You guys did awesome. Yay!